Hello, I'm Neil Spooner, the chair of the Patient-Centric Sampling Interest Group, and I'd very much like to welcome you to our webinar for March featuring Andrew Patterson, who's from Eurofins Environment in North America. Today he's going to be talking to us about advancing the collection and analysis of PFAS in blood. But before we move on to Andrew, just like to share this slide. Without our sponsors for the Patient-Centric Sampling Interest Group, we wouldn't be able to do these webinars for free. And please remember, we've got 11 of these webinars booked for this year. We had 5 last year. We've got already 4 booked for next year. And all the webinars that are coming, well, the ones for the next couple of months are on our website. The historical ones, the free recordings are all available on our website. So if you're interested, please take a look. But thank you to all our sponsors who are featured on this slide. So the agenda for today is, after I've finished filling your ears, Andrew's going to be talking to you. Please submit your questions through the chat function. Don't use the private chat, use the chat function. We'll be able to see the questions there, and we'll put those to Andrew at the end, and he'll be able to work through those. Please note the webinar is being recorded, so it will be available forever after this event is finished. And at the end of the event, I'll do a quick wrap up. So with that, I'll finish speaking and I'll hand over to Andrew.
Very good. Let me just get my screen pulled up here. Can you see my screen?
Yes.
Wonderful. Good morning, good afternoon. My name is Andrew Patterson. I'm the VP of Innovation and Development for Eurofins Specialty Services. Really what that means is I get to work with some amazing scientists and very high-end instrumentation, mostly in North America, to come to bring you solutions around PFAS and around legacy contaminants and other POPs. So let's just dive right in. We're going to have to move pretty quickly. This is the only structure in the entire presentation, I promise. Today, we're going to cover some PFAS background, but briefly, we're going to talk about how complex it is to test for PFAS. We're going to look at the approach that we came up for PFAS in blood, and we'll talk about what's coming up next for biomonitoring. So really quickly, PFAS, or per and polyfluoroalkyl substances, is not a new term. And this was a recent map published about all the presumptive sites that will have PFAS. I put some funding up on the right there, really not just to dive into the numbers, but more to see that there is a lot of dollars flowing into PFAS. And this could be anything from remediation research, destruction technologies, biomonitoring like we're doing, just general monitoring. So the full gamut all around PFAS. And that's, we think that those numbers are going to be dwarfed in the future by even larger sums. So follow the contamination, and the contamination is being treated very seriously. So we're at Eurofins. We have a network of independent laboratories within North America. We have several labs. I believe it's up to 7 now that support different PFAS matrices. And so we're a PFAS hub, if you will. So all these matrices we in fact support for PFAS analyses, and in some cases we're the ones writing the methods. Today we're just going to talk about blood and serum, but again we're talking about blood and serum because there are PFAS in almost every facet of your life, although that is decreasing these days. So PFAS in blood, really it's not if, but how much. I can tell you that I started my PFAS career in human serum in 2007, and I have yet to see a non-detect. So it really is not just it's not just chin music. There really is PFAS in almost every single one of us. So how do those PFAS get into your body? I really like this graphic because it shows you are being inundated from the air you breathe to the food packaging you might have to the food you eat, although food is pretty low, to the water that you drink, and everything else in between. And then you're also contributing, to be noted that you're contributing back to the landfill. You're contributing back into the surface water. So really PFAS are this vicious cycle. And there are some efforts being done to mitigate PFAS in everyday life. But the lion's share of it is already out there in the environment. So once PFAS are in your body, what do? Well, really depending somewhat on the chain length of the compound in question, they can remain in your body for a matter of hours or for years or for decades. On the top left there, that's PFBA. That's the C4 carboxylic acid. And you can see that the half-life's 72 to 81 hours. So if you were to stop being exposed to that chemical now, within 72 hours, half of it would have been either excreted or metabolized. Contrast that with something like PFAS or PFHXS, and we're talking years, if not decades, to go through even just one half-life. So it's part of the reason they're called forever chemicals. They exist a long time and are very stable in the environment. And in fact, they are fairly stable and exist for a long time in your body. So back in 2022, the summer of 2022, the National Academies of Science came out with their advisory. And this was really the first time that a government agency said, yes, you should test your water for PFAS, and yes, you should test your blood for PFAS. And so what they recommended was you do get your blood tested for PFAS, and then you take these 7 PFAS out of all of them that can be tested, and you add those. And depending on where you add up, they sort of divvied it up into 3 different categories here. Less than 2 nanograms per mil, and that's really not a whole lot of us. 2 to 20 nanograms per mil, and that's a lot of us, or most of us, and then greater than 20 nanograms per mil. And with that comes different risk and health effects. We're really not going to talk too much about the health and risk effects, but more on the technology side of things today. But I did want to point out when I talk about the fact that less than 2 nanograms per mil is not most of us, the fact that if you pull up the tables from CDC and Haynes themselves, and you just pop up the geometric mean for the total population for PFAS and PFOA, you can see that we're somewhere in the 5 point something region. So most of us already just from 2 compounds are going to be above that that low level background. I pulled up a couple more tables here and just wanted to illustrate that it's not just about the total concentration of PFAS in your body, but also the distribution. So a normal US distribution, and honestly, from what I've seen, every sort of geographic region should have their own databases to pull from because the patterns are different. But if you're in the United States, your PFAS should be higher than your PFOA, which should be higher than your PFHXS. And if that distribution is skewed or twisted, if say your PFHXS is the predominant compound in your body, that's atypical. And so there's been some exposure in your past that is not representative of typical US exposure. And that's also interesting. Most of the PFAS databases for blood are in serum. Serum is a little easier to work with, or has been traditionally. And I've just listed out some pros and cons of serum and whole blood. So we do support lots of serum samples. In fact, we support more serum samples than we do whole blood samples, but whole blood samples haven't been readily available for that long. And so they are growing. A few call outs for whole blood. It is easier to collect and it is easier to ship. We can ship in ambient conditions. And at the end of the day, your body pumps whole blood. It doesn't pump just serum. So I like to think that it's a little more representative of what your PFAS body burden might be. The CDC list is on the left and our list is on the right, our longer list, which does encompass the CDC list, but also has some legacy, some replacement chemistries, and kind of goes further into carboxylic acid chain length and whatnot, just to better serve really the research community. We do a lot of work with universities, and with them, just like it is with regulators, the more compounds, the better. The CDC list is shorter, but it's great. This isn't a dog on the CDC at all. They do fantastic work, and in fact, we're trying to mirror what they do with regard to linear and branched isomers, and we'll talk about those compounds in just a little bit. So PFAS biomonitoring, layers of complexity to get this done. PFAS are everywhere. I've just popped a couple of pictures up on this slide, really showing that all the places that PFAS background can exist in the laboratory, from the solvents that we use down to the liner and the caps of the solvents that we use, the water sources. That's a picture on the top right there of some HVAC systems. HVAC has been known to be off-gassing some PFAS, especially shorter chain PFAS. The standards that we have used to be not quite as pure as they are now, which is good news. Pipette tips, different SPE media and glassware can all have PFAS on them. And in fact, humans can as well. You yourself can be a point source by some of the products that you would use. So we have to be looking from every angle. And really, the take-home message for all of this is that PFAS blood testing really needs quality control umbrellas. Because the very components that we can have would bias the results and cause our false positives, we need to have negative controls in place, or samples that don't have any PFAS in them. Follow your samples around the lab in order to ensure that any detections are coming from your sample and not from the laboratory itself. I hate slides with lots of writing on them. That's why I made this one, because I don't expect anybody to read this. I just wanted to lay out everything that goes into before we analyze any samples and what we do on a regular basis to analyze samples. So this is being recorded. If you want to come back and read all of this, you're welcome to it. Just know that PFAS testing we take very seriously, and the QA QC is very, very rigorous. So in less words than that, defensible data is the summit of a mountain of effort. And really, since we're a PFAS lab first that's accredited for PFAS, and we have our PFAS reputation to write on, everything that we do is fully defensible. And we've been doing this a long time, and we like to think that we're only getting better out of it. So when you get your report, just know that that really is a lot of effort behind that report that you can't quite see. So how do we approach PFAS? Well, traditionally, PFAS in blood, you would get a phlebotomist visit. They would do a blood draw. They would generate serum. That is where you let the blood clot first, and then you centrifuge after that to separate out the phases. We can deal in plasma. If you have a project that you need plasma delivered on, we can also support that. But typically, more typically than not, it's serum, and that is because most of the databases are in serum, so they match up. But after you generate that serum, you do have biohazardous shipping that does have to be temperature controlled. So we thought serum works, but it's logistically complex. And what if there was a better way? And I'll tell you right now that during the beginning of COVID, we had several blood projects for PFAS that were canceled because we simply could not get into these areas that we needed to sample. So what I did during quarantine was work on the remote blood test. This has the benefit that there isn't a phlebotomy cost or inconvenience. We can ship at ambient conditions. We like to think that for children, this is an easier approach because the test can be administered in their own home by their parent or a caregiver. And we do try to consider costs because we do a lot of work for research. And so we like to think this will result in a greater number of participants for a research project. So in short, think a home DNA ancestry test, something like that, where you would order a kit, you'd get the kit at your house, you'd have everything you need to sample, and you'd drop it back in a FedEx mailer box that shows up in our lab in Sacramento, where I'm based out of. And then in a couple of weeks, you get your results. So the device you saw on that previous slide being sampled there is in fact the Neoteryx Mitra VAMS sampling device. It's a mouthful, but VAMS really just means the volumetric absorptive microsampling. And we'll get into the volumetric aspect in just a little bit. We're not the only ones using this device. It's brilliant. In fact, the inventor of the device recommended I come on this webinar and speak because I hopefully of how well it went. So this can be used for a wide variety of biological fluids. We're in fact using capillary whole blood. And a couple considerations for us is that the media needed to be volumetric and it needed to be PFAS free. We can't have PFAS already on the device if we're looking to measure PFAS. And that's my wife's hand right there. She's been sampled a couple times. On the VAM's accuracy and precision, this is one of the tables we're using to first bring up the device to gravimetrically determine how much volume it was pulling up each time. And long story short, we were, for a 30 microliter mitra, we were averaging 29.8 microliters with a 1% inaccuracy, and it was very consistent. We only had a 5% RSD. So this was great news. It's both PFAS free and it's volumetric and it's consistently volumetric. So for us, this was the fourth device that I tried during method development. So it was quite a relief to find it. Once you put PFAS on this device, the whole blood dries. We want it to dry. In fact, there's a desiccant card in the specimen bag. And so the blood acts like this great substrate. On some of our stability studies, I just pulled a couple PFAS out here. The green line was mitra kept at room temperature, and the blue line is mitra kept in the freezer. And I guess for the most part, it's hard to distinguish those two lines, the room temperature from the freezer. So it is pretty amazing. And we're not storing these at room temperature once they get back to the lab, but you can see that it's a pretty rugged device and a pretty kind of neat concept. So once your samples have the blood on them and they're at the lab, what do we do? This is a highly simplified approach, but we're using a lot of isotopically labeled PFAS, and we'll talk about that in a second. But we add the tips, we add our standards and the reagents to the tips. We're doing a sonication effort there. After sonication, we're doing SPE or solid phase extraction. That really removes some of the phospholipids and other bulk interferences that we would find in whole blood. After the SPE phase, we are going to spike in our NIS to use 1633 language. That's our non-extracted internal standard. That allows us to determine the efficiency or the percent recovery of the isotopes that we spiked in here, our extracted internal standards. We then analyze the sample. These are all reviewed by multiple real humans for their accuracy, and then we issue your report. There was a question about the sample size. Rightly so, people are concerned that with a microsampling device, that can we trust this? Can we trust these data? And so before we even dive into the validation efforts, I did want to point out that our old serum test, the test that we were doing at Eurofins back in 2019, 2020, used 250 microliters or a quarter of a mil. The new test, we're using 2 of the 30 microliter mitra tips for a total of 60 microliters of blood. When we set this up, we decided that actually that volume of whole blood works so well with our workflow that we actually matched our serum test volume-wise to the mitra test. And kind of ancillary to that is that now we can better deal with samples with limited volume. So we've done some maternal serum projects. You can imagine how valuable those samples are. We're doing paired mother-baby dyads where we have very limited volume. So if we're only using 60 microliters, that really opens things up to researchers. We've had researchers send us 120 microliters. We've used 60 and shipped the other 60 back. So pretty cool. So the take home really is that the volume of the new tests is about 4 times smaller, but our instruments, the new instruments we have, the SIAC 7500s, are safely 10 times more sensitive. So we're not saying we can do everything in the sun with a couple drops of blood, but we are saying that we can do PFAS pretty well with a couple drops of blood. Mentioned earlier adding isotopes to the samples. So why use isotopes? And by the way, these are not radioactive. These are stable isotopes using carbon-13 labels of PFAS themselves. So the benefit here is that they really do mitigate false positives and false negatives. And I'll take a second to describe what you're seeing on the right there. So on the instrument itself, the target compound, or the native PFAS in question, has two different ions, one that we measure the quantitation on and one that we qualitatively need present to assure that what you're seeing is in fact a positive. And then on the bottom there, you can see that we have 13C4 PFOA. So right away, visual cues, if this compound was not there, you would know that the test worked because you can see that isotope with confidence. And then, so that's protecting against false negatives. And then false positives, you can see if this retention time didn't line up exactly with the isotope, you'd see that right away. So it can prevent false positives. And then even better than that is the fact that if there's any suppression or enhancement of the PFAS, that they're going to be loss correct. That same suppression or enhancement will be happening to the isotope in question. So the data are in fact loss corrected, and you can have, you could in fact spill some during extraction or cleanup, and you'd still derive the correct starting concentration. We're a huge proponent of isotopes. We're using all that we possibly can. I would talk to whatever PFAS analysis you're looking to get done. Talk to your laboratory, not only ask them what native compounds they offer, but talk to them about isotopes, figure out how many they're using. This is also the most expensive way to approach it. But for us, this is considered the gold standard of quantitation. So we're always going to be adding isotopes as they become available. Isomers, you might have heard this called branched, linear, and total. And you'll remember from the tables earlier from the CDC that for some compounds, for some PFAS, they're looking for the L version or the linear version and the BR version or the branch version. So think of these like Legos that you would take apart a certain Lego, put the same number of pieces back on that Lego, but it might have a different shape. That's kind of like what isomers are. And in fact, the isomers have different retention times than the linear isomer. And so we can distinguish these on the instrument. I should point out that in the environment, we do encounter different mixtures of both linear and branched isomers. And as a result, in our blood, we accumulate different ratios of linear and branched isomers. And at the end of the day, your PFAS body burden is the sum of the linear and the branched isomers, but not just one or the other. Again, your body is pumping around the linear and the branched isomers at the same rate all over your body. So you've sent your sample to the lab. We've extracted it. You're all experts in chromatography now. We get to issue your report. This is a snip from one of our reports, and we do tease out the CDC and Haynes analytes on the top of the report. This has a couple columns that I want to draw your attention to. Really, the long spelled out name for many of the compounds, and then your result as measured in whole blood. This reporting limit is how low that the instruments can detect with 99% confidence a positive detection. And then we have our serum equivalent here. And this is all in nanograms per mil or parts per billion. So we're deriving these serum equivalents from peer-reviewed published papers. We did generate one of our own from a recent cohort, but more of these are needed. And the unfortunate part about that is that we need to find impacted communities that we can run paired samples of venous serum to capillary whole blood. The serum equivalent, I'll call your attention to one more time, because if you do have one of our reports and you're looking to compare your data against the CDC and Haynes, you're always going to want to use that serum equivalent. We are summing the 7 compounds we talked about earlier to get your NASEM total value. In this case, this is a made-up individual, but they could have a very real concentration of the sum of those 7 of about 46 nanograms per mil. That's the value that you then take and plug into the National Academy of Sciences ranges and kind of see where you pop out. So we're kind of working backwards now. How do you know that any of what I just said is real at all? And you should be asking that. We did publish a peer-reviewed study back in May 2023. This was published with Eurofins, Michigan State University, in conjunction with Duke University and also Colorado School of Mines. This was a 50-person cohort up in Parchment, Michigan. You never guess what they do up there. A lot of paper coding. We really did try to approach this from a really rigorous direction, in that we did paired venous whole blood, venous serum, and capillary whole blood, and pitted those against each other, because this we really did try to stress this to see where it worked and where it didn't. We did have the capillary whole blood collected by the cohort individuals with Mitra. So the serum samples, both whole blood and venous serum, were collected by professionals, and the capillary whole blood was collected by the participants themselves. There are some pretty astounding levels, and you can read about it more in the paper. But we did, for PFAS, we reached 120 nanograms per mil. And for PFOA, it was above 200 nanograms per mil. This was actually above our initial calibration range. And so it was a shock to see some of these levels, that we actually had to dilute the samples after the initial injection to get the concentrations within our concentration range. Tables, again, that I don't expect you to read, but part of the initial validations are those positive controls. And so throughout the study, our positive controls were coming back for serum and for VAMs in a really nice, comfortable range, with 88 and 99% accuracy, with not too much variation, hovering around 8% or 9%. Again, what this tells us is that if there were PFAS present in these samples, that we could detect them and detect them accurately. So how do the blood components compare? I pulled two tables here, and apologies, they do seem to be a little fuzzier than I first thought. I pulled two tables here from our publication. The one on the left is capillary whole blood compared to the venous whole blood, and I had to turn my head there a little bit. And really, this is a one-to-one line. And so that tells us, because capillary whole blood really is a mixture of a couple different bloods, but venous whole blood is specifically venous whole blood. So do those match? And we determined that yes, capillary whole blood and venous whole blood do match when it comes to PFAS concentrations. And then we had to determine, does capillary whole blood, is it predictive of serum concentrations? And that line that's plotted there is really the 2-to-1 line. So for the select PFAS that we pulled out there, you can see all the data line up really well. So yes, that capillary whole blood is predictive of serum concentrations. One thing though that previous literature had also published on is that there are some differences in distribution with PFAS. So I pulled a table from our supporting information and highlighted fossa down there. And as you can see, if you look, the 0 there was a 0% detection frequency for serum, and both for the venous VAMs and the capillary VAMs, we had a high 40-something% detection frequency. So 47% detection frequency for capillary VAMs. So if we were just looking at serum alone, we would have missed the fossa. But since we were looking at multiple types of blood, we caught it. And this has been reported before, but it is always interesting to see. And so, why would you care about fossa? It's not on that National Academy's list or anything like that. Well, it's in your body, and in fact, it can transform into PFAS over time. So it can act as a reservoir for future PFAS. So this was, I think this is this component, as well as some of the data on branched isomers, is some of the most interesting things about PFAS in blood. This slide really does follow up on what we were observing here. So 0% in the serum, 47, 48% in the whole blood. Why is that? This was the paper that came out in 2017. It's a fantastic one as well. And really, I've highlighted the fact here, this is the distribution difference, the ratios between serum and whole blood. So when we're above 1, the distribution is driven towards the serum. And when we're below 1, the distribution is driven towards the whole blood. And you can see here for fossa, that's 0.2. So that's about a 5-to-1 ratio of whole blood to serum that fossa wants to exist in. So that's what we were observing before, has previously been published on, which is great because then we can describe what we're seeing and use that coefficient. Along with our validation, we always like to include SRMs, their standard reference materials, wherever possible. One of the most famous and widely used is NIST SRM 1957. This is on human serum, and really we approached this 2 different ways. We absorbed a little bit of the serum with our VAMS device, and the serum technique was pipetted like we would for live samples. And you can see that, in fact, the VAMS, although a little more accurate, this was a little less precise than the serum approach, but still both sets were very agreeable with our team and others. I will call some attention to PFUNA. This was right down near the detection limit of our study. And you can see that both the VAMS and the serum approach are predicting that low level positive pretty well. We did find additional PFAS, and NIST asked us to publish whatever we found. And so we did find and published on 4 additional PFAS in that NIST serum, even though there aren't recovery values there because they haven't been published on enough. So go find these guys and publish on them. It's going to help the whole community out. That's like a call out, right? Okay. So what comes next with PFAS in blood? I think one of those things is going to be occupational testing. When you first say this, people kind of take a breath and they're not quite so sure about it. But if you think about it, we were already getting drug screens and background checks when we show up to a business. Imagine you're working in an industry that has PFAS around it. You get your background check, your drug screen, and perhaps you get your PFAS baseline. Because if you show up with a highly elevated PFAS baseline on day number 1 of your job, well, that's not from your current employer. That's from your previous, you're a composite of your lifetime of exposure. So that protects the employer. Now, to protect the employee, you should have personal protection equipment, or PPE, that is protecting you. And so your levels shouldn't be going up. So not only are you going to get a baseline test, you're going to get some follow-up tests on a regular basis to ensure that you are being protected. So I like to think that PFAS testing is going to protect both parties equally. What comes next? International PFAS in Blood launch. I'm very pleased to announce that our Japanese colleagues have taken our kit, our PFAS exposure blood kit, and are running with it. And in fact, there does seem to be a lot of interest in Japan. And this is, I've never been part of an international business collaboration, but this has gone great so far. And we're just over the moon that our Japanese colleagues are bringing on this test and that there's increased interest in PFAS testing. And I can assure you that I haven't seen a country yet that doesn't have some level of PFAS in it. Speaking of, this is at least a year underway now. There was a factory in Antwerp, Belgium, that was producing lots of PFAS, and that factory's was shut down for a while. I think it might still be shut down, but it seems like there is elevated PFAS background in Antwerp. And so our European colleagues reached out to us because they knew we had this test. We helped bring them online. And since then, Eurofins in Europe has launched the largest European biomonitoring PFAS project ever. This is a big deal. Recently, there was some news blurbs because, not surprisingly, when they give this test to different members of the parliament, they're finding lots of detections. Politicians are people too. So it's not surprising that they have PFAS in them, but it does tend to make the headlines because then the issue becomes very, very real for people. So between the Japanese launch and the ongoing Belgium biomonitor, you can see that the groundswell is here, and it really is here for whole blood. Although with the Belgium study, it is being collected both in whole blood and serum. We were so hyper-focused on human applications of remote PFAS testing that after webinars like this, someone would say, hey, can I do bird blood with this approach? And the answer is yes. Our instruments don't know the difference between all these different blood matrices. And our method really is set up for whole blood and serum and plasma. But we can do all sorts of whole blood and serum and plasma. So we do have projects underway for avian, rodent, and fish species, and who knows what else is out there. If somebody finds a Bigfoot, I'm happy to test Bigfoot's blood for PFAS. That would be groundbreaking. I'd like a special call out to a non-human application, which is a recent publication by the University of New Mexico. You can get to the paper by clicking that QR logo. And I did, in the middle here, put up a map, and it might look like an abstract painting at first. But really, this is the Holloman Lake, which is on the corner of Holloman Air Force Base. I think we all have an idea of how massive an Air Force Base is. This also kind of butts up to the White Sands Missile Range. Never know what happens there, which is also next to the White Sands National Park. And in Holloman Lake, the researchers did it was a really interesting paper. And we were fortunate enough to be associated with this publication. I just took a snip from one of the tables showing mammal liver. This wasn't blood, but this was mammal liver, and that's 97,000 nanograms per gram. So that's in ppm levels. That's 97 ppm. That's very, very high. So the title of extraordinary levels of per implied fluoroalkyl substances in vertebrate animals at a New Mexico desert oasis is not an exaggeration. And with that, I see that I'm coming up on my time limit, and it's also the end of my presentation. So with that, if you have any questions, please email myself or my colleague, Taryn McKnight.
Thank you, Andrew. Thank you for a fascinating presentation. I'll just see if I can share. So please, we've got people submitting questions. Please do so via the chat function, not via the private chat, via the shared chat. So I'll just read out some of these questions for you, Andrew. We got one here. Who is responsible for confirming if the future lots of VAMs are PFAS free? That comes from Hara Hisanori.
That's a good question. So if you're performing routine PFAS analysis, you should be having an ongoing quality assurance program. So all the media that we use are in fact QC'd for PFAS, and in our negative controls and our method blanks, we are putting the current lot of VAMS devices in with our controls. Good question.
Thank you, Andrew. I apologize if there's noise in the background. We're having our bathroom done, so I will mute in a minute. We've got a question from Russ. Hi, Russ. Do you use the 2 miter tip VAMS configuration and not use the second meter tip to measure any contamination in a blank meter device? So contamination from manufacturing or in the field, to ensure that your assay is actually from the person and not contamination.
Another good question. We're actually deploying a 4 meter device, and that's because we ask people to fill all 4 of them. So we collect 4, we analyze 2 filled tips for our primary analysis, and then we save 2 in the background. Because we're QCing our tips on a regular basis, and we're QCing the mailer bags down, and we're QCing down to the stickers that we use, we're not having an extra field duplicate or a field blank with all these samples, but it's something that we could consider if you had a project and you needed that. We're somewhat flexible.
Yeah, it'd be interesting to know whether it's environmental contamination or whether it's real from the blood. So interesting question coming here from Adam. Is there a difference between free versus total for PFAS? Could this make a difference in effect in the relative guidance levels? What about accepted levels for tissue exposure and any particular organ that is known to have high levels?
So the on the first question, I'm not quite following free versus total PFAS. On some of my slides, when it said total PFAS or total PFOA, that means the sum of the linear and branched isomers. And we, if you meant total PFAS, you want to mute a little bit.
I think what they're meaning is, so a lot of molecules in circulation might bind to serum albumin, alpha-1 acetylglycoprotein, things like that. So I think they're just asking, is there a difference?
Undoubtedly, there are other PFAS in your bloodstream that are binding to albumin. One of our first steps is that we perform a protein crash, and we're liberating, we'd like to think, all PFAS from those proteins. And remember, we are adding our own analogs, our own isotopes to that sample before we do anything else and letting it equilibrate. So that when we do protein crash everything out, we're liberating those PFAS. And again, we're only looking for a targeted list of PFAS, the list of compounds that we're looking for there. We are in early stages of discussing non-targeted analysis of PFAS in blood. This would be using either a QTOF or an Orbitrap high resolution instrument, so that we could look for, people say, all the stars in the sky type of thing for PFAS. But it, that would be, and some people are doing that, and it is very interesting, but it's not quantitative.
And that's really interesting that you're looking at that. I think the free versus total is an interesting discussion, because in drug development, we always measure total drug, or we think we do, but actually people are interested in free drug because the stuff that's bound to albumin and other plasma components is thought to be not biologically active. So I just wondered whether anyone had done any of that kind of work with PFAS, because it could make a difference to what we think of as harmful and not harmful, if a lot of it is actually bound up and therefore not hitting receptors in the body.
And I think that would bring up questions about equilibrium, with different PFAS. And so for us, we're really trying to exhaustively extract everything that we can. We're never just looking for the free. We're always trying to get to that total, or the total that happens to be in your blood at the time.
And the second part of that question was about, are there any accepted levels for tissue exposure, and whether any particular organ is hit? I think it was with some of the animal data, you've got liver data there. Is there any of that kind of data for humans, and are there levels acceptable?
I don't know of specific organ data for humans. It would be interesting. We have been asked to look at biopsied tissues, and it's something that we can do. Again, we're looking at all sorts of different animal tissues, and if we happen to get a human liver, we could absolutely run that through our tissue extraction process, which is different than our blood extraction process. But I don't know of any guidance for specific tissues, and I honestly don't think it's been studied very well at all. The only guidance that I've heard of is around blood.
Thank you, Andrew. So moving on to the next question from Vinicius. Have you optimized the extraction procedure at any level? More specifically, is there a reason for combining both sonication and shaking instead of just using one of the two? Was it a protocol suggested by Neoterics?
So we've really everything that we're doing extraction-wise is in that paper. It's great when we can publish. I work on a lot of kind of secret things where we can't say how we do it. I would say that if I had my druthers, I would just sonicate things. I think it's a little more aggressive approach than shaking. But we have done both in the past. That slide you saw was somewhat of a generalization of what we would do.
Okay. There's another question from Adam. can you mention where exactly in the sample extraction process you add the PFAS stable labeled internal standards for the meter tips, e.g. in the extraction solvent solution, or is it in the addition later in the extraction process? And I think you did cover that in your slides.
Well, but I do want to point out that we do spike the isotopes directly onto the meter tips. We just let those soak.
Thank you. Another question from Russ about longitudinal testing, commenting that that's an interesting idea. I'm just wondering how you differentiate between PFAS accumulation solely from your workplace when you can't do a null hypothesis experiment.
You can't, I think that's where some of the nuances in linear and branched isomers are going to come into play, or for novel detections. We cast a pretty wide net with our list of 45 PFAS, and a lot of those are non-detects. But if we run into a population that does have unique exposure that's outside of typical exposure, we'd like to think that we can capture that exposure by including as many PFAS as possible. But you're exactly correct. If you were exposed to some PFOA from your water, some PFOA from where you grew up as a child, some PFOA from whether it be some different fluorochemistry in your life, rain gear, makeup, whatever it is, at the end of the day, that's all commingled in your body. So you can't really point at one source until you start finding the nuances that we're looking for.
So, I have before we go back to some of the other questions, I have a question myself. If you come back, if someone comes back with high PFAS levels, what does that mean to them? What's the follow-up? What happens to them?
Well, honestly, the follow-up, the first follow-up is, can I have my water tested? And we do a lot of water testing as well, or people say, can we do remote air testing? And we do have some air testing capabilities already in place. So, which is the right thinking. So you are a composite snapshot of your lifetime of exposure of PFAS, but you can look to your current environment to see if you're still being exposed. So looking to your water, looking to your food, looking to maybe your textiles and clothing are a good first place. But honestly, it's already too late. So if your water is clean now, that means perhaps it wasn't always clean, where you grew up, or perhaps you had an occupation where you had exposure that is not typical of a general exposure. And that goes back to that changing the rank order of PFAS, or having anomalously high results for a specific couple of PFAS. And we do see that. We see where, PFAS to PFOA to PFHXS, the PFAS to PFOA is about 2 to 1. We've seen people that have PFAS that's 20 times higher than their PFOA. And you'd say, okay, in the past, I've had a predominant PFAS exposure. Where did that come from? And more often than not, you're not going to know.
But if you do get the high level, can anything be done about it?
So that's another interesting question, and I have some slides on a different presentation that talks about this. The 3 best ways to excrete PFAS from your body are by menstruation, lactation, and childbearing. So that cuts out about half of us. And there was a recent study on Australian firefighters where they were donating plasma, and they were donating whole blood. And they found that over a year of donating the blood, and I believe that they discarded the blood, they didn't put it in someone else, because firefighters have notoriously high PFAS body burden compared to civilians, that it statistically did lower the PFAS in their bodies. So we've come full circle with bloodletting.
Cool. Okay, interesting. Question from Ahmed here. Did you have a dedicated instrument, or were you able to use the MS for other applications? He says the PFAS I used were very sticky in the system.
So right, we do have dedicated blood PFAS instruments. In fact, we're pretty proud to get the first 2 SciEx 7500s, and to use those for PFAS and blood. You do need a really sensitive instrument. We have over 40 LCM SMS in our Eurofins Sacramento lab, and that's growing all the time. So we're lucky enough that we can be specific with what we do with regard to PFAS. And yes, again, I can't stress enough that you don't want to just jump in to analytical chemistry with PFAS.
Yeah, it's everywhere. Question from Cyprian. Do you recommend hand washing prior to blood collection?
I do. And in fact, I helped with my colleague Taryn and some of our other colleagues. We helped write the instructions for use, or the IFU. And so if I was to, and hand washing is the first step. But if I was to say what I think are the best steps for success for sampling with the Mitra, it's really, washing your hands, and do it with warm water so you get that blood flow. And then also to stand up while you're sampling. When you're standing, your hands, and keep your hands as low below your heart as you can. And that's going to give you the best chance at blood flow. Really, people seem to fall into a couple categories, kind of less to average or average blood flow, and then we have others that have a lancet and they could fill 6 or 7 or 8 tips. And so that's part of the game. If there was an easy way to use a swab with this test, I would be developing it. But right now we need just a little bit of blood. So it's this field is constantly advancing, though, and I think we'll get to something more advanced than whole blood, probably. But this is a big step forward.
Has anyone looked at other matrices, urine or hair, for historical exposure or things like that? Urine obviously shows an accumulation and excretion over time. Hair, people sometimes use that to get a historical record of what's happened in the last few months. Has anyone done that kind of work?
Definitely folks are looking at urine, and also looking, urine in paired studies, mostly with serum. Urine does show a different excretion profile, and can capture some of the short chain compounds that are transient, like PFBA. I can't think of somebody looking at hair right now, but I guess it could be done. I've done hair analysis for dioxins and PCBs in my past, so I do know it's possible, but I don't. When you do a hair digestion, it's really aggressive, and so I do think folks would rather use something that's a little more easily extractable, like blood or like urine.
Yeah, it's interesting. And hair easily contaminated, I guess. What's in shampoo and conditioner?
Well, it sounds weird, but I wanted to do earwax at one time. But then I was thinking that, things are going to fly and stick to the earwax, and then it's contaminated itself.
And people do nails as well, obviously, and things like that.
Yep.
So it's interesting. And I think this is the last question we have, unless anyone wants to post anything else. We've got one from Robert asking, how representative is the blood concentration with respect to total body load? Would it be worth looking at in tissue and in fat in animal samples?
I'd love to. And in fact, that last study I put up there from New Mexico, they were looking at paired blood samples to liver samples, to skeletal muscle samples. So that work is being done. I do think that, at the end of the day, for humans, that's a little more difficult. You'd either have to work off biopsies or necropsies. So, but it is, it would be interesting to look at the concentrative effect of some of the different organs. And we know already that the liver is one of those, but, I would be curious, is this showing up in elevated levels compared to other organs?
So last question from Russ. I'm going to let you sneak one in, Russ, seeing as it's you. How much do we really know about the toxicity of these contaminants when there are tens of thousands in the environment?
That is the $20,000 question right there. So the toxicity knowledge base is growing every year. And really, I know of some interesting studies, just because you hear short chain PFAS like PFBA. I know of some interesting studies on renal function in PFBA that are going on right now. And that's just one PFAS. But your question, your comment on how do we know, what the toxicity of some PFAS are when you're exposed to tens of thousands? We don't. Right now, I can tell you that our aqueous test is growing to over 100 targeted PFAS compounds, and that's because that's about every analytical standard that's out there. So, in 5 years ago, that number was dozens less than that. So as more analytical standards become available, then we can target for those compounds, and then we can use them as individual markers for toxicity. So really, we need as many standards as we can, and we'll be adding them as they're available.
And are they all in one assay?
What's that? The 100?
Yeah.
Yeah.
Wow, that's cool.
It is cool.
That's, we all know how difficult sometimes it is to get 3 or 4 analytes in a single assay. To get 100 in there that's in an LCMS method. That's brilliant.
Yeah. So that's the state of things. We used to congratulate ourselves back in 2005, 2006, 2007 for just PFAS and PFOA, and jump forward to now, and we're cracking 100.
Cool. No, that's really exciting. So I'd like to thank you, Andrew, for a really interesting presentation and a great Q&A session. You got thank you to all our attendees for putting in some really interesting questions here. And we had a great discussion. Just like to share a couple of slides before everyone disappears. And I can see people are already disappearing, so that's okay. Just remember that this is being recorded and will be available both on the evidence base and on the patient-centric sampling interest group for forever. Just a quick plug for next month's webinar, which is going to be focusing on sports drug testing ahead of the Olympics coming up in Paris. So we've got David Cowan, who ran the drug testing lab for London 2012, and Daniel Eichner from the University of Utah, who is involved in drug testing in sport in the USA and globally. So please take a look for that on the 23rd of April, and please register for that if you're interested. For those of you that don't know about the patient-centric sampling interest group, please, we're available. We're a closed group on LinkedIn. Anyone can join to just discuss all things patient-centric sampling. Our website has a lot of resources. There's some listing here of the kinds of things that we're doing. We are based upon people giving their time and resources and their passion to help us get things done, to help just move the barrier on getting patient-centric sampling processes more widely understood and more widely accepted. And again, I'd like to just thank our partners who enabled this all to happen today. And I'd like to finish by thanking everyone, and thank you, Andrew, for a fascinating webinar, and thank you to the audience for great questions and a really good discussion today. Really
great questions. Thank you very much. Thank you for having me.
No, you're welcome. It's fun. Thank you all.